Hey everybody, welcome back. Today we're going to lead into somatosensation. One thing that I've done with today's lecture that you'll notice that's different is I'm going to put in a part 1, 2, and 3. A few people have let me know that the lectures can feel a little bit long and it's not really clear where you could take a break and take a little bit of a rest to go off and do something else, go get a snack, eat some lunch. So anytime you see a title slide back up like this, then you'll know that this is a subsection that you could take a break with. Some are going to be longer or shorter, but I've tried to at least group some common topics together to help give you a little bit of a heads up for when that happens. So I might try that for the next few lectures, and we'll see how it goes. And if it works better, then we'll keep it, and if it doesn't, we'll get rid of it. All right, having said that, let's dive into the good part. So we're going to be talking about somatosensation today. And this is really the term that we use to talk about any signals that arise from the body. And so for all of you, the first thing you probably think of with this is your cutaneous senses, right? This is the sense of touch. And that is probably the primary component of somatosensation. But we're also talking about a number of other signals that arise from the body. And these include proprioception, which is our understanding of where our body is in space. And some of us are better at this than others. Some people walk into chairs and things sometimes, and other people can avoid them. And so having an awareness of where your body is and where all the different parts of your body are is known as proprioception. We also have kinesthesia, and this is the perception of how we move. And so this is really more of like the interaction with the body and its feedback, because of course, the sense of touch we think of, almost in a way where you're running your hands along something and you're feeling texture and or something bumping into you. But we also have perception over the way that our body is moving and how much strain is on a muscle group and things like that. And so we talk about that a little bit here and a little bit in the motor system. And then the last thing are the organic senses, and these are senses from the inner organs of the body. And so this is where sensations of kind of, feeling bloated or having something feel kind of warm or cold or even feeling sick. A lot of these things come in. So we're going to talk about all of those things today. So if we just break down the part of touch that we call cutaneous sensation, then there are multiple subcategories of this. So we have receptors that can detect hot and cold, some in the skin that are specialized to stretch, others that are responsible for detecting things like vibration. And notice that these are very close to the top of the skin. These are probably the ones that we all feel activating accidentally. That gives us the phantom phone vibration sensation that many of us are now accustomed to. And then we have specific receptors for pain and others that will help us to detect when we have movements of the hair follicles. And all of these send separate but kind of parallel signals out to the brain. So I already said this idea of kinesthesia and proprioception is just us having an understanding of exactly how our body is moving and also where the different parts of our body are in space. And this involves the integration of multiple senses and systems together, but a lot of this information comes through our somatosensory system. One component of this information comes from the stretch receptors that are at skeletal muscles and tendons, so this is giving us some information about the strain on the muscle. We also have information coming from the muscle length detectors, so these intrafusal fibers, and this tells us whether or not a muscle is stretched out or condensed. We've already talked about both of those, but of course now we have to add into this the idea of receptors on the skin. This is information about what the skin is doing at any given time. And so we have all these different sensors we just talked about in the cutaneous sense, and those provide information. When we move, our skin moves with our muscles. It makes pretty intuitive sense. So the last sense we need to talk about that falls under this category of somatosensation are these organic senses. So we have a wide variety of different sensors in our body, and the name for this category of internal sensors are interoceptors. So intero for internal and septors for receptors, and these give information for so many different things that can go on in the body. We have some that describe our temperature, and these are very similar to the ones that are in the skin. We also have some that detect whether or not there's some sort of chemicals or toxins, and these provide information that tell us when to vomit or when we have too much salt in our body or any number of other things like that. Heart rate, so you can all tell, you become very aware of your heartbeat at certain points in time, but it's not all the time. If your heart's beating day in and day out, we're very much habituated to it. And yet, if your heart's racing because you've had too much coffee or you've run too hard, you instantly become very consciously aware of your heart rate. Metabolic function, so we have internal sensors that tell us things about when our system is too bloated, we have too much food, and those are internal stretch receptors. And we also have sensors for the chemical composition of what we've eaten. And we'll talk about those a little bit more in a future lecture on ingested behavior. And then, of course, blood pressure, right? So we think of this as being how dilated our vascular system is, our veins and arteries. So vasodilation, we also have sensors for. We can also kind of tell when our blood pressure is really, really high, and we have a lot of changes going on related to that. All right, so let's just take a minute and walk through all of these different receptors in the skin. So one very important thing in understanding how our cutaneous senses work is understanding first the anatomy of the skin, because our skin acts like a filter for many of these different cutaneous receptors. Our skin has 3 layers. Right? We have a layer of subcutaneous tissue, and this is the lowest layer. Then we also have a middle layer that we call the dermis, and you've maybe heard of this before, it sounds familiar, but the one that most people are familiar with is the epidermis, okay, which is the top layer of the skin. So these 3 different layers become the location where we house some of these cutaneous receptors. So in looking at the receptors in the skin, there are 4 different kinds of skin receptors. So what I'm going to do is I'm going to show you where these are and talk to you a little bit about them. And then I'm going to describe to you the theory behind why we might have so many different skin receptors, so that as we go through them, you can take a look and potentially understand why we have these different classes of receptors, all to do something very, very similar. So the first of these are these Merkel's discs. You can see these here. And one thing that I want to point out about these is that they are actually these small pink nodules and not the larger white blobs that you're seeing. The second of these are the Ruffini corpuscles, and these Ruffini corpuscles are slightly deeper in the skin, but still kind of right at the border between the dermis and the epidermis. The third of these skin receptors are the Meissner corpuscles, and these are actually those white blobs that you might have been looking at before. And then the last of these are these PACINIAN corpuscles. And these are located very deep, pretty much right at the layer, right at the border, rather, between the dermis and subcutaneous layers of the skin. So there's a bit of a thought about why we have these different receptors. The main idea here is that these different classes of receptors, when you boil everything down, are really all started out from the same kind of receptor in the skin, and that they evolved over time. And they really evolved in 2 different ways to change their sensitivity. So a good way to think about this would be the same way that the retina has the pupil to kind of open and close, but the skin doesn't have that ability. So instead, it developed a few different classes of receptors. Some of these have capsules around them, like these Meissner's corpuscles. You can see they have this large kind of capsule around them, or the PACINIAN corpuscles, that they're embedded in some other very specific kind of tissue that changes the way that they perceive things. And others, like the Merkel's discs, really are not. So if you take this and you put it together with 2 other factors, how deep something is within the skin. Is it at the epidermis or is it the dermis? Is it encapsulated or is it not encapsulated? And then all the way at the level of the bipolar neurons that these connect to, do the neurons respond quickly or do they respond slowly? Putting all of these factors together gives you the ability to have different levels of sensitivity in the system. So before we move on, I also want to point out that there are two other types of sensory receptors. The first of these are the free nerve endings. So the free nerve endings here, you can see come all the way up and pretty much into the epidermis, much like the Merkel's discs. And then we also have the hair cells, so specific sets of cells that wrap around the hair follicles and allow us to detect whether or not there is movement of the hair. All right, so let's look at the details of these different skin receptors, and all along the way you should be thinking about those three factors. Are they encapsulated or not? Where are they located in the skin? And then do they respond quickly or slowly? And to just give you some idea of kind of the important thing to know here is you should understand how these different skin receptors relate back to those jobs we were talking about. They either respond to stretch or light touch or vibration. So go back a few slides and look, if you need to, at the way that these Merkel's discs play a part in the different things we think of as being a sense of touch. So these are located on hairy and hairless skin. And they're very superficial, which means that they have very little skin protecting them or filtering information from reaching them, depending on which way you want to look at it. And they also happen to be unencapsulated because remember, we're talking about these small pink kind of nerve endings here. So these don't have a large capsule around them, and they respond to light touch. But then we bring in this third factor, which is the type of neuron that they're connected to, which happens to be slow adapting. So they're sensitive to light touch, but they're not rapidly responding. So they have pretty good sensitivity, but maybe they're not the fastest message we get to the brain. Now let's look at the Meissner's corpuscles. So remember, these are these white blobs over here, but interestingly enough, they're only found on the glabrous skin in small elevations in the dermis. Okay, so glabrous skin, remember, is the skin we just defined as being that skin that has no hair. And these are also fairly close to the surface of the skin, but they have very small receptive fields, which means that they respond to a very small surface area on your skin. And they respond to fine touch as well as very low frequency vibrations. Okay, so let's put these pieces together and think about where we might be likely to find these. So where can you think of on your body? It's a place that responds to fine touch and actually does that very, very quickly. So incredibly high sensitivity in both time and space on skin that has no hair. Think about it for a second. You have an idea in your head? If you don't, look down at your hands. So your hands are one of the few places where we don't have hair. And so these are also tools that we use for all of us because they have such high sensitivity to touch. In fact, they're so sensitive that it's almost a place where you can't experience touch that feels very pleasurable. It's almost like it's too sensitive of an area of your skin. This is why we get really irritated, especially as all of us are applying hand sanitizer 1,000 times a day or washing our hands 1,000 times a day. When the skin gets dry or anything else, it's very irritable because it has such high sensitivity. So these Meissner's corpuscles, that's primarily where they're located, or in places like the bottom of the feet or the hands. And these are encapsulated, which might seem counterintuitive because they have this big pad around them, but what actually it does is it allows them to take information from more of the skin, and that capsule for these seems to act as some kind of an amplifier. So now let's look at these Ruffini corpuscles. Okay, so these Ruffini corpuscles are located on hairy and hairless skin, and they're located just like the Merkel's discs, very superficially. And they actually, even though it doesn't look like it in this picture here, they're encapsulated, but they have these very small capsules. And what they seem to respond to is stretch, as well as sustained pressure. So one thought about this is that this has something to do with the way that these are grouped. And a piece of evidence that supports this is that their responses, when we think of stretch, are directionally sensitive. So these Ruffini corpuscles seem to have a specific arrangement in the skin. They're in some sort of a line or a matrix, and they respond maybe something like the hair cells in the ear, where movement in a particular direction seems to activate them. And much like the Merkel's discs, they're fairly slow adapting. So it seems that as we get to these smaller corpuscles and skin receptors, that it takes a lot to cause a change in them. So again, this relates somewhat back to the way that they're formed, and also has something to do with their location in the skin. The last of these skin receptors are these PACINIAN corpuscles. So they happen to be the largest receptor in the skin, and they're also the deepest of all these skin receptors. They're located on both the hairy and the hairless skin. Pretty much everything, with the exception of these Meissner's corpuscles, is located all over our body. As you can see here, especially if you look at this one in the middle, these PACINIAN corpuscles have a weird, almost like an onion-like structure to them, where they have a lot of layers. And they also have this big padding around them. So somehow this all translates to these having a very large receptive field. And yet, much like the Meissner's corpuscles, these have a very rapid response, and they respond to high frequency vibration, and also to just regular touch. So putting this all together, I think you can see that each of these has a slightly different job, and they act almost like every other system that we've seen, where we have different receptors that are tuned in different ways, whether it be in the retina to different colors, or even things like differences in location. Now we have all these skin receptors that have evolved over time to respond to different kinds of touch that we might experience, probably all from some common type of skin receptor. So now let's talk for a second about these other sensory receptors that we brought up. The first of these were these free nerve endings, and these are found in both hairy and glabrous skin. So before I tell you more about these, I want you to do a small experiment. And you're probably sitting at home on your own laptop or phone listening to this. So there's nobody here to judge you, so play along for a minute. Something I want you to do is that I want you to just take your hand and try and caress, for lack of a better word, your forearm or someplace with hair. Okay? And now I want you to do the exact same thing, try and use the exact same motion across the palm of your hand. And then just think for a second about how both of those feel. So these free nerve endings are found in both hairy and glabrous skin. But in the hairless or glabrous skin, they are actually competing with these Meissner's corpuscles that we've already said are really high sensitivity. So the free nerve endings, their primary job is to be high sensitivity, that's why they're near the surface of the skin, for two things. Temperature, so hot versus cold, and then also damage. So these free nerve endings are our primary receptor for painful things, especially painful heat or cold, and tissue damage that activates inflammatory factors like cytokines and things like that we haven't really talked too much about, but they're chemicals that signal inflammation. The reason for our experiment is that in the hairy skin, these also seem to play the exact opposite role and are responsible for pleasurable touch, this feeling of very positive touch. So they're incredibly sensitive receptors, and they play this juxtaposing role in both pain and pleasure, to make the cliche out of it. But that's one really interesting thing about the way this system seems to work, and this only happens in skin that has hair on it. Like I said, probably because of competition with the Meissner's corpuscles when we get into places that are very, very high sensitivity, like the palm of your hand. And then hair follicle endings, or these hair receptors, as you might imagine, are found in hairy skin. Really hard to have a hair receptor on hairless skin. And they're located pretty much at the base of the hair follicles, and they seem to be a type of mechanoreceptors. So similar to the hair cells that we have in the ear, they're detecting movement of the hair in a way that's incredibly mechanical. So when the hair moves, we open up a receptor due to the direction of movement and so on and so forth. And these allow us to detect all kinds of different changes that happen when our hair moves. So next, let's take a moment and just discuss how we get the transduction of all of these different kinds of signals. So pretty much for all of the different corpuscles that we talked about, they have the exact same mechanism of action. So the detection of sensation by all of these is done by mechanoreceptors. So mechanoreceptors, you can think about, if you remember the auditory system, like these hair cells, where some sort of mechanical change, right? By this we mean if something is stretched or pushed or pulled by pressure or vibration or whatever it might be, that this causes a channel to open. And actually the channel is the same as we remember for those hair cells. Remember these tip links, they had they made a connection between two hair cells in the auditory system, and when those big columns of hair cells moved, then these trip channels, I told you to think of it like a trip wire, would pull the receptors open. And so the mechanoreceptors in our skin do the exact same thing. These use one of these trip channels. So in remembering these hair cells, these trip channels are simply ion channels that have some sort of connection to them. And when there's some sort of force on them that pulls and tightens them, then we can pull these channels wider open to allow the flow of ions in. That's it. All of these work in pretty much that same way. So whether you realized it or not, you actually already understood how the transduction for these would work. So when we look at the detection of temperature, the transduction here acts somewhat like we saw with the cutaneous receptors, where we've evolved to have a variety of different receptor subtypes that are specifically tuned to certain ranges of temperature. So if you look here, you can see this pretty clearly. We have a TRPM8 receptor whose peak sensitivity is about 25 degrees Celsius. And so this detects things that are a little bit cold, and somewhere around 40 to 45 degrees Celsius, we have these TRPV4 and TRPV3 receptors that are sensitive to warmth. And then as things get warmer, or to the point of hot and painful, we start to use TRPV1 and TRPV2 receptors. And then something that is missing from the table in your book, which I had copied over here, are actually a newer class of receptors for cold pain, these TRPA1 receptors, which we now know to respond when things get down to about 5 degrees Celsius. So with this different tuning, we are able to detect all kinds of different changes in temperature, because we have some things that respond to incredibly cold and other things that respond when things are incredibly hot, and pretty much everything in between. Another thing that I'd really like you to notice here is that these receptors are not just responsive to hot and cold, but the way that we discovered them is that they're actually also responsive to chemicals. So in the case of this TRPV1, they respond to capsaicin, which is what's found in chili peppers and other hot peppers. And the TRPM8 was actually named with this M for the menthol that it was found to respond to. And this is why if you put menthol on your skin in various different forms, or if you have a cough drop with menthol, then we feel this sort of coolness effect. And it's because these activate. Okay, so now let's do another one of these quick at-home experiments. So what I want you to do is to take a deep breath in, and I want you to blow at your hand 6 or 8 inches away. Okay, now remember what that temperature feels like. Okay, think about it for a second. Hold it in your memory. So next what I want you to do, and listen to the directions, is I want you to close your hand into a fist and breathe into your palm for 4 to 6 seconds. And then quickly do the same thing we just did right after you've done this. So breathe into your palm for 4 to 6 seconds, and then quickly take a breath and breathe at your hand. Okay, let's do it together. Breathe in, and then blow at your hand from 6 to 8 inches away. Take note of the way that you perceived each of those. The first time when you blew at your hand, and then the second after you've warmed your hand up by blowing into it. So if you think about this experiment, there's a few things that you should have noticed. For us as mammals, we have cold sensors that are located superficially and warm sensors that are deep in the skin. And what this does is it prevents false positive triggers. When we start to get cold, we feel it, but what we want to do is make certain that our internal temperatures remain consistent. And so changes in those warm sensors can activate things like shivering. And that means that we have placed them appropriately for being warm-blooded. The other thing that you should have very quickly noticed is that feelings of temperature are relative. And the exceptions to this are for those sensors that sense extreme cold or extreme heat to the point of being painful. But otherwise, we're looking for relative changes in temperature for the things that might trigger any responses for these temperature sensors. And then what this allows us to do is to adapt to new levels of warmth or cold. And so this means that if you're outside in the cold for a long time, but it's not painfully cold, then a room is going to feel much, much warmer when you first come inside, and then we'll adapt to the warm room, and going back out to cold will feel much worse. And this allows our body to kind of modify its responses as need be, depending on the environment. So I think you all kind of get the idea here. Another thing that's important to notice that differentiates some of these TRPS receptors for hot and cold from some of the other sensors that we have in the skin is that these receptors also respond to chemicals. So the TRPV1 receptors that are most important for making something feel kind of painfully hot respond to capsaicin, which is what you find in really hot chili peppers. You may also, for these TRPM8 receptors, remember that they're responding to kind of cold, and in the case of these, they respond to menthol, which comes from mint. And this is why we perceive some of these things as being having kind of like a cooling or a warming sensation, and they're often used in all kinds of chemical compounds. And interestingly, these TRPA1 receptors respond to mustard oil. And so there are a few others that are kind of interesting. So the TRPA1 will also respond to cinnamon and garlic, and there's an entire kind of range of different plants and chemicals that these will respond to. When we think about receptors for pain, then anytime we're talking about a receptor that's responsible for detecting pain, we call this a nociceptor. These detect what we call noxious or negative or unpleasant stimuli. And there are a few different classes of nociceptor. So the first is that we have high threshold mechanoreceptors. And so these respond to mechanical stimulation, but it has to be something that's very forceful, like a pinch or when your hand or a part of your body gets hit. The second that we have are these heat-sensitive channels, right? The trip-sensitive channels, and they respond to both heat, but part of the importance of them responding to chemicals is that there are also some noxious chemicals that cause damage to the skin. So that's very, very important that we have a class of pain receptors that responds, of course, to heat, but is also responsive to many different chemicals that can cause inflammation and damage. And then we have some that respond to other classes of chemicals. For example, the TRPA1 we said responds to extreme cold, but there are also chemicals there that can cause inflammation. And so this seems to be a commonality of a couple of these different channels. And as I said, the main point of these TRPA1 and V1 channels is having really a general system for pain that responds to anything that can create either inflammation, in the case of the TRPA1 and V1 channels, or tissue damage, which will eventually result in inflammation. So in one case, it's a little bit more direct because things like capsaicin can cause inflammation almost immediately and act on these receptors. But in the case of something like injury or tissue damage, within a few seconds, we start to have the activation of inflammatory signals. So we talked very early on about responses to pain as being one kind of an example. And basically all that we're adding here to that example of kind of picking up a hot mug or a bowl of soup or anything like that is one of the initiators of the response. So now we know we have these free nerve endings out in the skin, and they can respond to something like heat in the case of the example that we used. And these become the receptors that start this signal, as they are going to send a signal into the dorsal horn of the spinal cord, and they pass by an interneuron, or in some cases a series of interneurons that we talked about as being very, very important for being able to modulate these kind of responses, because these would get a top-down signal from the brain. And then simultaneously, these signals will trigger a muscle response, which is something we'll talk about a little bit more later, and simultaneously send a signal to the brain so that we can have an opportunity to perceive that something has been painful and act accordingly to take some sort of a future action and prevent ourselves from burning our hand on a hot stove again, or to start to take care of that wounded limb if need be. One sensation that we really haven't talked about much yet that falls under somatosensation is our sense of itch. And I wish I were kidding here, but I'm not. Itch is scientifically defined as some sort of sensation, obviously something from the outside world, or that comes into contact with our skin, or a sensation within the skin that causes the desire to scratch. So in one of the weirdest and most circular definitions we will look at it this entire semester, an itch is literally a stimulus that causes us to want to scratch. The sensation of an itch is caused by a chemical that is in the skin and that's released when one of these stimuli that from the environment causes us to want to scratch. And you've probably all heard of this because many of you have taken antihistamines. And so the activation of the chemical within our body called histamines is what causes this sensation that makes us feel the need to scratch whenever there's some sort of an irritation to the skin. So one of the coolest things that you never knew is that the reason that we like to scratch and itch is because there is this teeter-totter or balance yin and yang to pain and itch. So pain can actually reduce itching. So if we scratch so hard that we actually produce something kind of like a pain response, we can block the itch response. And in complete contrast to that, if we add something that creates a sensation of itch, we can reduce pain. So a really important thing to know is that we are still kind of on the hunt for the receptors that control this sensation of itch. And yet the idea is that these are probably also some sort of receptor that's on the free nerve endings, and that there's likely completely contradictory responses for when something generates a response that we perceive as being itchy, and a different set of receptors would be activated for a response we perceive as being painful. And for whatever reason, these two seem to counteract each other when they hit the brain and the spinal cord and so on and so forth. So we don't fully understand these yet, but one of the interesting and cool things we have discovered is that these two work against each other. The next thing that we're going to talk about are somatosensory pathways. So if you remember, for each of these sensory systems, the three things you really need to know are how the signals are transduced and they're sent to the brain, the pathways that those signals use, and then some information about the functions that this transduction and that these signals are able to support. So in part 2 here, we're going to be talking about the pathways, but this is just your reminder and nice stopping point here to where if you need a break, this is probably around the halfway mark. For this particular lecture, there's a lot to talk about in this first section. So if you need a break, hit pause here and you can go out and take a walk for a couple of minutes or do something to give yourself that mental break you need, and then dive back in. One of the things that we're going to be doing in this part of the lecture is that we're going to be comparing and contrasting the systems that we use for pain and for other components of somatosensation, because there are some important differences between the two. So for both of these systems, then we have nerve endings that are found out in the skin, and these could be the free nerve endings that perceive pain, or these could also be the different kinds of corpuscles that are involved in the transduction of various kinds of touch, like vibration or soft touch or anything of that nature. And these neurons are, much like the ones in the visual system, a kind of bipolar neuron. So one pole for these neurons is out in the body, and the other pole comes into the spinal cord. And in between them is the actual neuron, right? Represented by these two dots here, right? So these neurons, these bipolar neurons, are found not in the spinal cord, but directly adjacent to the spinal cord in a grouping of neurons called the dorsal root ganglion. Okay? It's just this tiny cluster of neuronal cells and that you see where those dots are. So the axons containing information about touch, they ascend through a part of the spinal cord called the dorsal column, and you can see that here where the arrow indicates. The neurons that are coming through this dorsal column, they travel all the way up, essentially to the brainstem, up to the medulla, before they finally cross the midline, and then they pass all the way up through the medial lemniscus and up to the ventral posterior nucleus of the thalamus in the brain. So one thing you should notice here is that these somatosensory neurons travel almost all the way up the spinal cord, and they hit the brainstem before they cross to the contralateral side. And another thing that's really important to notice here is that these neurons take a somewhat direct path in doing this, where the axons are coming all the way from these bipolar neurons in the dorsal root ganglion and up into the thalamus. From the thalamus, this information is relayed to the primary somatosensory cortex. So if you think about all of the systems that we've been talking about, then what we have here is fairly consistent with pretty much every other sensory system that we've looked at, where we have these neurons that are sending long-range projections, and they come through the brainstem and all the way up into the thalamus, and then they go on to the primary cortex and then on to a secondary cortex. So the pathways have kind of this parallel structure. There are some differences in a few of the pathways, especially those that are very, very close to the brain, but we have this fairly consistent structure. The pain pathways follow almost the exact same track, and we're going to be talking about just a couple of subtle differences, but these differences are important. So the cell bodies for these neurons are also located in the dorsal root ganglion, okay? And that is consistent with the somatosensory system. One of the major differences, though, is that these pain and temperature sensing neurons immediately synapse in the spinal cord, and that they also immediately cross the midline. And there are a couple of points to this, but one of the biggest reasons that this happens is that these pain neurons need to do two things simultaneously. So the first is they need to be able to have some interaction at the level of the spinal cord so that they can cause reflexive responses. So if you put your hand down and something is very, very hot, they need to be able to trigger a very rapid reaction from the motor system. And so that's one of their jobs. And then the other job is to inform the brain about what's happening. And so the signal that comes through this spinothalamic tract is the system that's informing the brain of what's happening. So once these neurons have crossed the midline, then they carry up through this spinothalamic tract. And you'll see if you compare this to the somatosensory system that it takes a fairly similar track, except that it's already on the contralateral or opposite side of the spinal cord from where the sensation happened. So this carries again all the way up through the medial lemniscus, and it ends up in the exact same location in this ventral posterior nucleus of the thalamus. Okay? So the systems become fairly parallel at that point. So much like in the somatosensory system, the thalamus is relaying this information about pain to the primary somatosensory cortex, which is going to process it and relay it on to the secondary somatosensory cortex, and so on and so forth. Another big difference between the somatosensory system involved in touch and the component of the somatosensory system involved in pain is what happens when these systems hit the brain. So the system for pain actually diverges very, very quickly. We've already said that this gets into somatosensory cortex, and this is what's involved in the sensory dimension of pain. So you could think of this as the most pure form of nociception. There's a painful stimulus that's come in, and we have to perceive it. But the other thing that happens is that these signals will branch into other parts of the brain that are involved in things like cognition, or our emotional response to pain, or even in generating a quick autonomic response by doing things like raising heart rate and blood pressure and so on and so forth. And we're going to talk a little bit about those next time and talk more about the functional aspect of this. But in just looking at the pathways, you should recognize that from the thalamus forward, that the pain system branches off so that it can start engaging our entire body in parallel to mount a response to this painful stimulus. So much like we saw in the visual system, the somatosensory system is incredibly organized. And the way that this is organized is somewhat similar to the visual system in that different parts of the body, much as we saw different parts of the visual field and different types of stimuli, are represented in different parts of the cortex. One thing that you should know, and I'm going to introduce you all to my ugly looking friend here, the homunculus, which is often this sort of representative character that's used for the somatosensory or the visual system, is that areas of the body that have higher sensitivity, kind of like us talking about rods versus cones, are going to have a greater representation. And the homunculus here is basically meant to be a visual depiction of that breakdown of representation for the somatosensory system. So things like the hand that we need for a lot of fine touch have an incredibly large representation in the cortex, whereas other parts of the body, like the arm, for example, have a much smaller representation. And this becomes very important for the way that we're able to have differences in perception for different parts of our body. And last but not least here, we're going to get into part 3, which is discussing a little bit more about pain as a part of this somatosensory system. So as you remember, our sense of somatosensation involves touch, but this involves a lot of different components that could be considered touch. And one of these components is pain. This becomes a very important component. We need to understand when pain hits our skin and other parts of our body. And part of this can even include hot and cold. So how in the world do we perceive pain? So if you think about pain just for a minute, it's probably one of our most evolutionarily advantageous systems. This allows us to look at things in our environment that are causing harm to us and allows us to learn about them and potentially avoid them in the future. It also allows us to heal by causing us to favor body parts that are injured. For example, if you've ever pulled a muscle or something like that, your body will favor that portion of itself for a short period of time to allow you to heal, sometimes longer periods of time. And we talked already just about the receptors that are responsible for this. We call these nociceptors. And just as a reminder, these are meant to detect any sort of unpleasant stimulus that we come in contact with. And this is usually some sort of inflammation-causing chemical or something that causes tissue damage that creates a painful response. Now, these are the sensations of pain, but the thing that we're focusing on in this last section is really the perception of pain. So one thing that you might be asking is why we have this disconnect between the way that we sense pain, right, through our nociceptors that send signals into the spinal cord, and the way that we perceive pain. And the reason for this is that there are multiple different systems that are involved in processing painful stimuli. The first of these, I think, is the most obvious. It's the one that we all think about. It's this sensory dimension of pain. This just tells us that some sort of sensory stimulus has come into contact with our body and tells us that the stimulus was painful and allows us to do things like withdraw the hand or foot or whatever injured component that there might be. On the other hand, there's these other two components that go together, the affective and cognitive dimensions. And we think about them separately in some cases, but the important thing here is to understand that these are involved in the calculated processing of how we perceive pain. So this is where the way that we're thinking about that painful stimulus can influence the perception of pain in a top-down way, and it can make us pull our hand or foot back even faster because maybe we're expecting something or we're not expecting something. Or when we get the affective or emotional component involved, that's where you can think of this idea that sometimes something that wouldn't normally bother you can bother you more if we're in a heightened state of emotion in some way or another. So if you are already sort of triggered and in a negative mood, then you might perceive something that is painful as being even more painful. And this is where sometimes when we look at things like chronic pain, they tend to be even worse in people that have depression or anxiety disorders, because these mood disorders affect the way that someone's mood might be at a given time, and therefore they actually end up affecting the way that people perceive painful stimuli. And this becomes a very vicious cycle where things are more painful and that makes things feel worse, and therefore that can worsen depression. And so it becomes this negative feedback loop that can be really problematic for chronic pain and mental illness. So how do we know that the brain plays some kind of a role in this? How do we know that it's influencing pain by way of emotion? Well, there are actually parts of the brain, and one of these is the insular cortex, where if you stimulate them, even though there's no painful stimulus coming into the body, you can cause somebody to perceive a an ambiguous, painful stimulus. They feel as if they are in pain, even though there's no pain coming into them. And actually, for individuals that have had some sort of a stroke or a brain lesion that affects the insular cortex, then they have deficits in a really important type of behavior called a nociceptive behavior. So an example of a nociceptive behavior is this pulling back of a hand very quickly, this rapid paw withdrawal. So if you think of what you would do when your hand touches a stove, that immediate withdrawal to move your hand away is called a nociceptive behavior. What these are really just defensive behaviors that we use to protect ourselves from a nociceptive stimulus. So the nocic in nociceptive behavior comes from nociceptive, and defensive comes from defensive. So this is a defensive behavior against nociceptive stimuli. So where does the cognitive side of things come in? Well, cognition is just the way that we think about things. And one of the ways that we often talk about cognition in psychology is this idea of top-down control, right? We have kind of the higher order parts of our brain, and these higher order parts of our brain are able to wrangle in the emotional parts of the brain. And that's why, humans, especially adults, are superior, because their prefrontal cortex has control. And that's a little bit of what happens. I mean, it's a bit of a cliche or a stereotype too. Lots of these prefrontal areas are involved in processing emotion. But if you use hypnosis and you tell somebody that the thing that they're about to perceive is going to be less painful, and you convince them of this, and they start using those cognitive elements of control, then we actually do see some sort of top-down influence on the emotional parts of the brain. If you tell somebody that they're incredibly strong and nothing can hurt them, then the emotional parts of the brain are going to have a smaller response to whatever the painful stimulus is. And the cool thing about this is it actually comes full circle, that if you ask somebody, rate for me on a scale of 1 to 10, how painful was this pinprick or this, hot plate that you had to put your hands on, they're actually going to tell you that it's less painful, but this carries all the way down to the somatosensory cortex. So there's actually some sort of almost like a gating where the stimulus that gets into the somatosensory cortex is lessened. So not only is this not just perception, but there are actually neural connections that are causing the somatosensory cortex to receive a smaller signal than it would have before. Another really interesting example of how pain and the brain interact in a way that's not purely just about the nociception or the sensory stimulus that's coming in is phantom limb syndrome. And if you haven't heard about this, one thing I would really encourage you to do is to go look at the work of Ramachandran, who is kind of the original person to spend a lot of time studying and even correcting the phantom limb syndrome. And so the idea of phantom limb syndrome is that when somebody has to have a limb amputated, there is still a lot of these free nerve endings that perceive pain. And so what happens is that there's a disconnect between the signal coming from these nerves that still have some connections and pieces and what the brain is doing. And the idea that Ramachandran came up with is that especially for people that might have a hand that's been amputated, that part of the reason they feel pain is because the signals are as if the person's clenching their hand into a fist and they can't let go, or something else that's causing kind of almost like a muscle tension perception in the somatosensory cortex. And what he was able to do is to trick the brain using something called a mirror box. And you can see the mirror box here, where what you would do is you would take the unamputated hand and you would allow a person to look at the unamputated hand, but then the mirror allows them to see something that looks like the amputated hand on the other side. And so as we've said throughout this entire process of looking at the different sensory systems, information from our visual system and our auditory system talks to the somatosensory system and the olfactory system. And what happens here is that we trick the brain into thinking that the amputated hand is still there, and that it's no longer holding this tense pose, or it's no longer perceiving this pain. And this treatment actually works. It causes people to do something that's like unclenching the fist that they're not able to physically unclench, because their brain now thinks that it's fine again, it's working, it's correct. And it causes them to have some relief for their chronic pain. And as it turns out, there's actually another way that the brain participates in pain, which is that it has its own internal pain management system that we call the endogenous opioid system. So endogenous just means native to the brain, and these opioids are a set of neurotransmitters that act on the same receptors that opioids, which you're probably all aware of because of the current crisis with opioid drugs. So these receptors are the same ones that morphine or heroin or fentanyl or other opioids might act on. And so it turns out that if you stimulate certain parts of the brain, we can cause them to release these opioids. And this causes something called analgesia, which is a diminished sensation of pain. So if you've ever had some sort of procedure in a doctor's office or a dentist's office where they numb either your gums or a part of your body to do some sort of minor surgery, the drugs that they use are called analgesics because they remove the sense of pain from that area for a period of time. And the brain can do this too. So if you stub your toe or something like that, the brain will try and release some of these opioids and help you to have some relief from that sense of pain. And it's not incredibly long lasting, but it's meant to help us manage pain for short periods of time. This is also the system that's allowed Olympic runners to continue finishing a sprint even though their leg is broken, and other things. Is the brain can jump in moments of adrenaline and other things because we're able to use this system when we need to for fight or flight and things of that nature. So what ends up happening when we stimulate a specific part of the brain, like the periaqueductal gray, is that we end up causing a reaction that goes all the way down to the spinal cord. So this is not just this opioid system is changing the way that we perceive this in the brain, it's actually changing the way that the pain is sensed down in the spinal cord. So what we think happens is that these opioids come in, and what they do is they cause disinhibition. So disinhibition is the idea that we have some kind of an inhibitory neuron that's being shut down most of the time, but we actually shut down the neuron that would normally be shutting this one down. Okay? And so this then allows a signal to carry all the way down to the spinal cord. We know that this goes through the raphe magnus, and we have this neuron in the dorsal horn of the spinal cord that might be involved in pain perception. And so what ends up happening at the end of this entire train is that we have interneurons that become activated, and they actually inhibit the signals that would normally be coming up to the brain from these free nerve endings that are perceiving pain. So if a stimulus were to come in to the neuron or the axon that's perceiving pain, this neuron's no longer going to send that signal back up to the brain through all the pathways that we've talked about. So this is a way where our brain can actually act to change the way that we even sense pain when we need to. It blocks all of the signals in kind of a top-down manner. All right, everybody, so that wraps things up for somatosensation. One thing I'd like to know, and you can drop me a note or put something in the chat, is whether you like this new format or not. So if you do, drop me a note. If you don't, feel free to drop me a note too. I just noticed in the introductory videos as I was finishing them out this past week that a few people said they would really like something like this, so I thought I would try it. With that being said, I hope you all have a great rest of the week. You know where to find me for office hours, and I will catch you next time.